Listen-only mode.
Hello everyone, and welcome to today's webinar. My name is Sherlyn Chen from Boehringer Ingelheim, and I will be your moderator for today's webinar that is organized by the GastroPlus User Group. Today's presentation is titled Human Exposure Predictions and Food Effect Risk Identification using Preclinical Data and PBPK Models. This presentation will be given jointly by two presenters, Dr. Tycho Heimbach and Dr. Wen Lin, both from Novartis Institutes for Biomedical Research, East Hanover, New Jersey in U.S. Dr. Tycho Heimbach is a director and lab head at Novartis. Currently, he chairs a global team which reviews and makes recommendations for formulation and PBPK PD strategies across all disease areas within the global DMPK function. He also serves as a representative on a global food effect and biopharmaceutic review team, where the main objective is to strengthen the biopharmaceutics and PK/PD sciences between different line functions at Novartis. Tycho has extensive experience with GastroPlus and PBPK models for human exposure and food effect predictions, and has authored or co-authored more than 15 papers. Dr. Wen Lin is a senior investigator 1 at the same company, Novartis. Her key responsibilities include PK/PD formulation and ADME studies for regulatory submission across all major disease areas, including application of pediatric PBPK models. Wen has extensive experience in both GastroPlus and other PBPK PD models, modeling preclinical and clinical data through all phases of drug development. She has authored or co-authored more than 20 publications. Also participating as a panelist today is Dr. Vera Lukchova from Simulations Plus. Vera is the team leader for simulation technologies and also serves as the GastroPlus product manager. Following the presentation, there will be an opportunity to ask questions. You may either send your question using the questions pane on your control panel, or you may ask your question directly using the hand-raising icon. If you are using a telephone to listen to the call, please be sure to enter the unique audio pin displayed when you join the call. This enables us to unmute your line so that we can hear you. This webinar will be recorded and will be available to the user group members in the future. So without further ado, we now begin the webinar presentation. Tycho.
Thank you, Sherlyn, for the kind introduction. Today we will share how we are using GastroPlus at Novartis across different stages of drug development, and also depending on the compound disease areas. So the presentation today will be, there will be 3 sections. The first one will be a review of our recent publication, which Dr. Wen Lin will present. We will also have some topics regarding our modified release strategies at Novartis, and then I will present on the assessment of food effects and share with you some discussions that we have at Novartis to assess food effect risks across all compounds and BCS and BDDCS classes. So this slide that you're looking here at sets the stage for us to talk about how at Novartis we use GastroPlus and modeling. So you can see the candidate stage where we all often charged with predicting our first human dose. We call it anticipation of human dose, the first one. This is prior to the IND stage. And at this stage we often have to use GastroPlus for the assessment of toxicokinetic formulations and preclinical studies. It is at this stage that we typically start with our human dose predictions that Dr. Van Lin will be talking about. Now, we also use GastroPlus at later stages. We use it for the food effect assessments prior to first-in-human studies. We use a GastroPlus PK/PD modules prior to first-in-human studies, and we use GastroPlus also for food effect assessments and general BA/BE type of criteria all the way through phase 4 and life cycle management. Now, on this slide, we are just highlighting how complex absorption and human PK scaling is. It is complex, but it is doable with tools such as GastroPlus. Because one thing what you're looking at here in the middle is you see a purple box and you see an orange box. The purple box is the 9-compartment ACAT model from GastroPlus, and the orange box there, which is systemic pharmacokinetics, is the clearance and volume of distribution. These are the disposition parameters that we also need to determine. And one thing we can say here is that in the publication that Dr. Van Lin will cover, it is important for the outcome of the modeling that you have a good understanding and good input parameters in both everything that goes into the purple absorption modeling box as well as what is going into the systemic PK parameter estimation. And I don't want to spend too much more time on this slide other than to say that we will be talking about the different input parameters that are going into the models. They're just as important as the model itself, because if any of the input parameters are incorrect or not biorelevant, then you can imagine that you can have errors in your predictions. So with this very brief introduction, I'll hand it over to Dr. Van Lin, who is going to be taking the lead to tell you about human dose predictions.
Thank you, Tycho. Hello, everyone. This is Van Lin from Novartis. And so I'm going to present the human PK prediction experience at Novartis. This slide shows the paper we published this year in Journal of Pharmaceutical Science. In this paper, we described how we use GastroPlus ACAT model as an integrated tool to predict the human PK profile for 18 compounds. This integrated tool we applied at Novartis can be applied across industry using the routinely generated in vitro and in vivo data. And this slide shows the decision tree for prospective PBPK prediction for human plasma concentration profiles, and which is also present in our paper in Journal of Pharmaceutical Science. At Novartis, we routinely fully utilize in vivo preclinical PK data. Based on the preclinical IV data, we predict the human clearance using allometric scaling method, FCIM, etc. We can also predict the human volume distribution using allometric scaling method or total method or two-species method. And then from here, we can predict the human IV profile using the Wajima method based on the predicted human clearance, predicted human volume distribution, and the observed preclinical IV data. As we know, for majority of the drugs, they are developed for the oral dosing regimen, not for the IV. Therefore, it is very important to estimate the absorption of the compound in human before we run the human trial. At Novartis, we have two methods to estimate the absorption of the compound in humans. For BCS1 compounds, usually we just use average F and the Ka value, and then combined with the predicted human IV profile, we can predict the PO profile for BCS1 compound. And then for BCS2 to 4 compound, in the prediction is for the absorption is more complicated because usually the absorption is not linear across the dose range. And we would like to use the animal PBPK absorption model, which is ACAP model in GastroPlus, to obtain in vivo relevant deconvoluted solubility. What's the purpose to perform this step? Because in most cases we notice the in vitro aqueous solubility cannot represent the solubility in vivo. There are many interfering or other factors can change the solubility of the drug substance in the presence of the formulation and in the presence of biosource. So that's why we want to obtain the in vivo solubility using the animal ACAP model. And after we set up the ACAP model in the animals, like in rats or dogs, and then we start to build the human PBPK absorption model, ACAP model in GastroPlus using the in vivo deconvoluted solubility together with the KCO2 permeability data. And then we can estimate the fraction absorbed in human at different dose levels, at different dosing regimens. Combined with the predicted human IV profile, we are able to predict the human TO profiles for BCS224 compounds. So I'm going to give the case example about how we utilize this decision tree for the human dose prediction. And so this slide is the prediction performance for 18 Novartis compounds using our integrated human prediction tool. All the solid lines are the predicted human plasma concentration profiles. All the circle symbols are the observed concentration profiles. So you can see for majority of the compounds, the fitting between the observed and the predicted profiles are very good. For majority of the compounds here, we use the GastroPlus ACAP model for the human PK prediction. Okay, case example, compound 1. For compound 1, we have preclinical data, both IV and PO data in rat, dog, and monkey. And so the first step is to predict the human PK parameters. We predicted the human clearance using allometric scaling method corrected by MLP. And since we measured the plasma protein binding percentage across species, so we predicted the volume distribution in human for this compound using oitoso method. And then the following step is to predict the human IV profile. The method we used is Wajima method. And the plot on the left-hand side is a very typical Wajima plot. So you can see the y-axis, which is the concentration axis, has been normalized by CSS, the concentration at steady state. The time scale, which is x-axis, has been normalized by MRT, mean residence time. Ideally, in the Wajima plot, and the transform or normalized concentration profile superimpose on each other from different animal species, which is present here in this plot. And then we can obtain the human PK profile, which is the plot here. Actually, to obtain the human PK profile, we need to do some back converting. So which means we need to convert the y-axis to concentration using the predicted human CSS, and also we need to convert the time scales to the actual time using the predicted human MRT, and then we can have this predicted human IV PK profile, and then you can based on the shape of the predicted human profile, you can go ahead to obtain the corresponding PK parameters. Okay, after we obtain the predicted human IV PK parameter, and then we can proceed to build a ECAP model for compound 1 in GastroPlus. The first step is to import the chemical structure of the compound 1 as a MOL file, and then we need to provide the human jejunal permeability in this box. You can use the Tycho2 permeability data and then convert to the human PF here. And then the second step is to select the absorption model. So we want to build a human absorption model under fasted conditions. So that's why we select the human physiology fasted model. And the AS model is by default chosen. And notice, GastroPlus provides the bile salt concentration in different segments in the gut. The bile salt concentration is about 2.8 millimole in the duodenum, and then along the from the upper small intestine to the lower small intestine, the bile salt concentration reduced to 0 in colon. So please bear in mind the bile salt concentration will affect the in vivo solubility of the compound. The third step to build an ECAP model is to enter the PK parameter in the PK tab here. So the PK parameters, they are derived from the previously described human IV profiles. So this is the two-compartment model. And before we run any simulation for the compound, we need to think about the in vivo solubility. And GastroPlus provides a dissolution model which can incorporate the bile salt enhancement on the solubility for the compound. So this slide showed how we use the bile salt enhancement function provided by GastroPlus to estimate the in vivo solubility for compound 1 in the ECAP model. So for this compound, we have the biorelevant solubility in FGF, facet, and the feces buffer. But we know we cannot input the solubility in the facet buffer as a reference solubility. It's not appropriate because we know in colon the bile salt concentration is 0. So if we use the solubility in the facet buffer, and then GastroPlus is going to estimate very high absorption in colon. And so what we did is that we used we turn on the bile salt effect function in GastroPlus, and GastroPlus can estimate the solubilization ratio for this compound based on the in vitro measured biorelevant solubility, and then this solubilization ratio output by the GastroPlus model consider the effect of the bile salts on the in vivo solubility in all regions of the GI tract. And for this compound, the duodenum solubility was estimated to be 4.3 milligram per ml, and actually GastroPlus also estimates the solubility for this compound in jejunum, ileum, and in the colon, and then accounting for the in vivo solubility at all regions of the GI tract, the ECAP model was able to simulate the human PK profile, which is shown in the plot in the middle here. And you can see this ECAP model can very well simulate the observed plasma concentration profiles in humans. That's one thing I want to mention. We have a high confidence to predict this human PK profile because prior to building this human ECAP model, we already built a dog ECAP model. This dog ECAP model already verified the in vivo solubility we use is appropriate to predict the dog PK profile. So that's why we have very high confidence to predict the human PK profile using the in vivo deconvoluted solubility. So this is the first case example. And in this slide, I summarize the PK parameter prediction methods we use at Novartis. For human clearance prediction, so we use 8 methods, including allometric scaling method, FCIM, and a single species method. For volume distribution prediction, we use 6 methods, including allometric scaling methods, or itoso method, and a 2 species method, and a single species method. And now I move to the second case example for the human PK/PD profile prediction. For the second case, the human prediction for AUC and T-half was less than 1.25, for which was excellent for the human PK prediction. And going to the detail for the human clearance prediction, we use allometric scaling method corrected by MLP. And for the human volume distribution prediction, we have a range of prediction using different methods, and then so we determine the volume distribution in human is 1 liter per kg. For this compound, we have the efficacy study, so we model the preclinical efficacy data, and then we use the rat EC50 from the rat model as the human EC50, just based on the experience of the front-runner compounds. And in the lower panel, there are 3 graphs. The graph on the left side is predict the human IV profile for this compound using the Wajima method. And then we continue to build the ACAP model to predict the oral profile of this compound. The graph in the middle is a prediction of the human plasma concentration profile as a green curve showing in this graph. And since we input the PD parameter from the rat efficacy model, so we are able to predict the human PD profile in this graph as a rat curve here. The graph on the right side is observed human PK profiles as a red line here, and the human predicted plasma concentration profile as the blue line here. And you can see the fitting of the observed versus predicted concentration profiles is very, very good. So this example further confirms the validity of the integrated tool for the human PK and PD prediction we described in our papers. And so here I conclude the first topic, the human PK prediction, and now I move to the second topic, how we use the modeling tools and animal study to assist the MR formulation development at Novartis. And this slide shows a paper we published last year. So in this paper, we're using dialtiazem as a model drug to quantify the regional drug absorption in human GI tract using IntelliCAP and the GastroPlus modeling tools. And this is a guidance for the modified release assessment tools, particularly for the IntelliCAP studies in human or dogs. The picture showing here is the IntelliCAP picture. It is a swallowable millipump. It can mimic all drug release profile for all MR formulations on the market. It is a smart IPO. It can measure pH and the temperature profile in real time, and it gives the readout to a laptop every 10 seconds. So the advantage of IntelliCAP are very obvious. The pH measurement on time can help us to determine the exact transit time for the IPO in stomach, small intestine, and colon. and combined with the plasma concentration profile, profiles either in human or in dogs, we can determine the absorption in each segment of the GI tract, especially the colonic absorption in dogs or human, which is very important for the NMR development. The limitation of the IntelliTab is due to the relatively small size of the drug reservoir, only 0.3 ml for liquid or 330 milligrams for powder. And also the relatively large size of the IPO may change its transit time in the GI tract. So at Novartis, before we run any IPO study in dogs, we need to set up a GastroPlus ACAT PBPK model to determine the colonic absorption or colonic availability. And actually, the cost to run a 5-dog IPO study were much less than the human study. So it is worthwhile trying to run a dog study before you want to run any modified release trial in human, which is very costly. And so this is the structure of the IPO. So IPO has about 4 major components: pH sensor and temperature sensor, a pump. So the drug release profile can be transferred into the IntelliCab or IPO memory before we give the IPO to dog or human. The reservoir here for storage of the drug substance or drug formulation in liquid or in solid form. So the IPO is made by Medimatrix, which is associated with Philips in Europe. This slide shows the typical in vitro drug release profile of marketed drug products. You can see the zero order or first order dissolution profile at different rates. IntelliCab can mimic oral release profile. And I mentioned before, the advantage of IntelliCab is it can determine the transit time in the GI tract in dogs or in humans. So this is a mechanism how it can determine the transit time in the GI tract, because IPO can locate itself in the GI tract by the pH profiles. So in this plot, the blue curve is a temperature profile of the IPO in the dogs. From the temperature change, we can determine when IPO was injected by the dog and when the IPO was excreted by the dog. The green line here is the pH profile in the dog. So from this plot, we can say this IPO stay in the dog for about 90 minutes, because at this point of inflection, the pH changed from 1.2 to 6 to 7, and then from this segment we can tell this IPO has stayed in the small intestine for about 150 minutes, because at this point of inflection, the pH drops from more than 7 to lower than 7. We know in cecum, due to the presence of E. coli, the pH reduced to 6.4 to 6.8, and then we know, okay, this IPO enters to the colon section, and then we can determine this the length of the IPO stay in the colon and the large intestine in the dogs. The next slide is the poster we presented in AATS a few years ago. So we used diltiazem as a model drug. The graph here is a pH profile of, and temperature profile and the plasma concentration profile of diltiazem in one human subject who ingested IntelliCap with a dissolution profile mimic the in vitro 24-hour dissolution profile. And the graph here is still the diltiazem in dog and human study plasma concentration profile. We model the data using GastroPlus in dog and human, and then we estimate the total absorption for this compound in dog or human was about 80%. And also we can estimate the canonic absorption for this compound in dog and human, which was about 60%. So which means this compound has very good canonic absorption in colon, either in dogs and in humans. So that's why it is suitable for the development of the modified release formulation. But keep in mind, on the market, only about 7 to 8% of drug has been developed into the modified release formulation, which means more than 90% of drugs are not suitable for MR development at very early drug development stage. And we can use this IntelliCap study in dogs combined with the GastroPlus model to identify which drug is suitable for the MR development and which drug is not suitable for the MR development. And we can stop the MR development for the drugs that are not suitable for MR development, because MR development is very expensive. And now I move to another decision tree, and Novartis, how we choose MR candidates and the site absorption studies based on BCS class, fraction absorbed, and a priori absorption modeling. So the commonly hold idea about the ideal drug candidates for MR development, they are BCS1 and BCS2. For BCS3 and BCS4, due to the post-permeability in the colon, so they are not ideal for the MR development. So for BCS1, the criterion here is, in the rat ADME study, the fraction of stop is more than 80%. Or in silico simulation, the fraction of stop is more than 80%, and also the metabolism is more than 70%. For the BCS2 compound, even the solubility is maybe low, but after formulation optimization, and then if the compound can give fraction of stop in the rat ADME study more than 80%, or in silico we predict the optimized formulation can give more than 80% of fraction absorbed in vivo, and metabolism more than 70%, and then we can consider this compound to be developed into MR form. And then we started to build the GastroPlus ACAP model for this compound. And if the predicted human PK profile using the ACAP model in GastroPlus is not desirable, for example, the short T1/2, and then we can consider the further MR development for this compound. Of course, this predicted human PK profile need to be verified by the clinical data. And then we can either conduct a conventional MR study in dogs, or we can conduct a site absorption study using the IntelliCap, as I described before, and then we use the dog in vivo study combined with the GastroPlus model to predict the human modified release PK and PD performance, and also colonic bioavailability in GastroPlus. I need to mention, so we need to have the end game in the mind, not only to predict the PK performance of the MR formulation in the human, we also need to consider the PD profile in the human given by the MR formulation for example, for some compound, even the PK given by the MR is not desirable, but maybe the PD can maintain the desired values in humans, so it's still suitable for the MR development, and then they can go ahead to develop the human modified release formulation by our colleagues in the formulation department. Okay, I think people are more interested in the case example, how we're using the decision tree for the MR assessment. So this is the first example for compound B, and this is a BCS2 compound, low solubility and high permeability. The safety margin for this compound appear to be low, possibly due to the high Cmax. So that's why we want to develop a MR formulation for this compound to minimize the Cmax-driven toxicities. So what we did is we estimate the colonic absorption of this compound using GastroPlus. So the graph here is a GastroPlus model. GastroPlus gives the output for the colonic absorption of this compound for only 4% in dogs, and then we conducted an IPO study. So this is a typical output by the IPO, the pH profile in dogs in the GI tract and the plasma concentration in dogs. And then we model this data. So the graph here is a GastroPlus model output. And so we estimate the colonic absorption for this compound in colon was only less than 10%. So the result showed this compound is not suitable for the further MR development due to very low colonic absorption. The second case example using the PBPK and IPO tools in dogs. For this compound C, it is another BCS2 compound, and the current dosing regimen in human is BID dosing regimen. The project team wants to switch to the QD dosing regimen using the modified release formulation. So we apply the same approach, and we run the IPO study in the dog, and based on the observed in vivo pH profile and the plasma concentration profile in dogs, so we are able to estimate the colonic absorption of this compound in dogs, which turned out to be about 40% in dogs. So this simulation results tell us this drug is suitable for MR formulation development due to the much higher colonic absorption in dogs using the IPO and the GastroPlus ACAD model. And so I conclude my presentation and then handed this back to Dr. Tycho Heimbach.
Okay, now we will switch a little bit in topic, and we're going to be talking about food effects and GastroPlus ACAD or PBPK modeling. To us, ACAD modeling is PBPK modeling, so we use the terminology interchangeably. At Novartis, we conduct absorption modeling for all BCS compounds. And the topic of food effect modeling assessment is relevant and timely. As some of you may know, there was a FDA, AAPS, ASCPT, ACCP workshop that occurred in February to evaluate if the current food effect guidance from 2002 is optimal and if it needs updating. So a number of things were discussed there. And it is not possible for me to capture all the discussions there. the food effect proposed revised guidance will be posted later this year for everyone's input. But I wanted to capture here some points that are relevant to a GastroPlus user community. And that is that FDA currently does not necessarily accept waivers for a food effect study just because of a BCS classification. So this means currently FDA and also other regulatory agencies are asking that we often run a definitive food effect study for the final market formulation. Sponsors are, of course, encouraged to provide modeling, but the message here is that there is no specific mentioning of PBPK modeling in the current food effect guidance. So before we talk about food effect, just making sure that everybody on the line is familiar with terminology. So a positive food effect, as shown here, denotes that the area under the curve is increased in the presence of food. This is typically the case for compounds that have poor solubility dissolution, where the solubility is enhanced by bile salts, and then you get this little bit delayed Cmax, Tmax, but you get an overall increase in the area under the curve. And of course, what that means is that in the fasted state condition, the bioavailability or the systemic exposure or the fraction absorbed was not 100%. And then similarly, the negative food effect does not mean that you don't have a food effect. It means that the AUC ratio is reduced. And I want to honor my late advisor here, David Fleischer. He actually was a specialist in drug transport and transport phenomena. And at that stages, GastroPlus was still in its infancy. But we are happy now that we have these new integrated tools that allow us to do these food effect assessments without having to write our own differential equations. I think anybody who works in formulation or in the MPK area probably is familiar with BCS classifications, class 1 to class 4, so I'm not going to go into great detail here. But other than to say, when you're looking at the food effects, there are significant impacts that are possible depending on the biopharmaceutics classification class. And what you can see here is for class 1 compounds, most of the time you would expect no difference in the extent of systemic availability. You can think of this also as bioavailability if you have an IV arm, or if you predict an IV arm, then you can call this bioavailability or bioavailability. But in some cases, we don't have this, so we're calling the systemic exposure, F extent. So class 1, most of the time, no huge changes, no surprises. Class 2, this is often dose dependent. These are compounds where you have solubility issues, dissolution issues, and most of them tend to increase, and this was also brought out at the FDA workshop. The class 3, these are ones where you have reduced exposure in the presence of food, and the magnitude of the reduction is actually very difficult to predict, even with current GastroPlus tools and current in vitro tools. So most of the time at Novartis, we can identify that there may be a reduction that can occur, but the magnitude is more difficult to estimate. And here the class 4, you could have put a question mark in the box because it is very challenging to predict, and the food effect can be dose and formulation dependent. We will share 2 examples here today. One is for class 1 and another one is for class 4. For class 2, you can look at some of our Novartis publications because we had a number of papers there that were also written with Bin-Feng Xia, who was our postdoc here. Now, just very briefly, switching from BCS to the BDDCS. So the DD stands for drug disposition, and the concept here is that to identify the proper BCS class, you can be using additional information to confirm whether or not you have more class 2 behavior or more class 4. If you have class 4, I have already explained that the certainty in the model is significantly reduced and that you can have surprises in the clinic. So for us, it is important to know if you are a class 2 compound or class 4. In the BDDCS, you use new information that is typically available for the compounds to identify the class, and that is the metabolism of the compound. So the metabolism of the compound determines the class, and what you basically do is, according to Leslie Bennett, you're replacing permeability with metabolism. So low permeability becomes low metabolism, and high permeability becomes high metabolism. And I will show you why that is important. We don't have time to go through the whole decision tree, but there was a paper from Binfeng Xia in 2012 where we described the decision tree depending on early discovery, candidate selection, or clinical development. And at different stages, we have different confidences about which class we identify. And again, knowing the likely BCS, BDDCS class is not a foolproof mechanism to have the prediction correct with absolute certainty, but the BDDCS class can help you identify that your confidence in the prediction is low, and it can point out to you that you may not be able to properly predict the food effect in humans. So at Novartis, we use metabolism information from the rat, so this is in vivo metabolism, to help us identify if the compound is more a BCS class 2 or more BCS class 4 drug. There are, of course, some assumptions here in the model, and but we do use this in vivo information in all of our models. So you can see here in this rat BDDCS from our rat ADME radiolabel study, if we find that after intravenous dosing that there's actually a relatively large amount of drug which is not metabolized, we're talking large amount means 30 to 40% is intact, which is basically as what you dosed, no metabolism occurred, then we say this is a BCS class 4 drug. Compounds that we think are high metabolism or translated to high permeability compounds, if you give them intravenously, they will show a high degree of metabolism, and you will see less intact drug. And again, we published this particular decision tree. So the decision tree purpose here is to identify how the compound might be behaving in the clinic based on this simple classification system. Again, this is not absolutely foolproof, but it is suitable to identify the risk. So here you are familiar with permeability and solubility on the BCS. So high permeability, high solubility is class 1, high permeability, low solubility class 2, and so on. So up to this point here, it's just the BCS, and we have certain numbers that we arbitrarily assign to separate out high versus low. But the metabolism piece is the one which is providing the new information here. And again, we're using the rat in vivo metabolism to identify or classify the compound here based on high metabolism as either 1 or 2, or poor metabolism as 3 or 4. And this information is then important also for our assessment of when we should be using additional dog studies and what type of modeling approach we will be using with GastroPlus. So this decision tree here focuses on BCS, BDDCS class 2 drugs. And the BDDCS, BCS 2 drugs, the positive food effect identification is a little bit more straightforward than class 3 and class 4. So this decision tree assumes that the compound has a big solubility differences, whether or not you give a buffer without bile salts, and then compare the buffer with which has lecithin and taurocholate, and then you compare the solubilization ratio in these two buffers, And then you also look at the dose number, because higher dose number compounds will tend to give you incomplete absorption due to limited dissolution. So in this decision tree, we can identify high-risk compounds. So for all of these compounds, BCS2, we routinely conduct GastroPlus modeling for the human outcome. And in some cases, in a few select cases, we also conduct the dog pharmacokinetic studies with formulation screening, because we want to make sure that the formulation that we're bringing into the clinic is reasonably optimized for high absorption in the fasted state. So ideally, at Novartis, we will already have an enabled formulation going into the first patient, first visit studies. And if it's a class 1 compound, we might just run GastroPlus modeling. But if there's questions, we will do GastroPlus modeling, and in a few cases, when we cannot take any risk because we're moving very quickly, we will run also a dog PK study. We do not tend to run dog PK studies of BCS class 4 drugs, and I will come back to that. So now I mentioned to you this FDA workshop, food effect, and you would think, well, then maybe you don't need to worry about if you have a class 1 compound. Well, not so quick. While BCS class 1 compounds probably are not as resource intensive in terms of the overall formulation development, prediction of food effect is not recognized by everyone as easily doable. So here we have a class 1 compound with very high solubility and high absorption as established by high bioavailability in preclinical species. And we also have a high predicted and known bioavailability in humans. And then I mentioned this metabolism in the rat after IV dosing is mainly metabolized, also indicating potential for high permeability, and there are no absorption or complexation formulation issues. So then you would anticipate that there should not be any food effect be expected, because if you imagine if the fraction absorbed in the fasted state is already very high, above 80%, if you then give this food and you have a BCS class 1 compound, the exposure can only go up very marginally, and there's no mechanism listed here that would explain a negative food effect. So here are the data for a compound here on BCS class 1 compound. In this case, we would have the predicted fasted and observed fasted data, and I will submit to you that with the latest PBPK GastroPlus model, the generation of the blue curve is relatively straightforward and can be conducted with high confidence. The issue here is that apparently there's not enough data consistently compiled across different users and companies to have a strong argument to convince regulatory agencies, and we have to understand their concern, because they're looking out for efficacy and safety for all drugs across all disease areas. Our point here is that this study actually was run several years ago. In today's day and age, the blue curve would be predictable very well with the latest tools. And the way, of course, we use the GastroPlus, we have FASIF and FESIF solubility, but we also have this human fasted data, and the paradigm would be that if you have human fasted data for a BCS class 1 compound with high bioavailability here and high fraction absorbed, you should be able to predict this fed state performance with high confidence. And what we're also showing here is the parameter sensitivity analysis. That is one of the major advantages of PBPK modeling, where we're showing that the AUC ratio, fed over fasted, is predicted to be constant and not changed as a function of a wide dose range. Now, this compound has very high solubility, so you do not see any increase in AUC ratio here. But most compounds, even if they have lower solubility, if they're BCS class 1, the line should still be unity. And this can be readily done using GastroPlus parameter sensitivity analysis. And in the interest of time, I'm going to go to the next slide. These are just the numbers. The second example that we have after this BCS class 1 drug, this was a compound that was originally classified as 2, and so you will remember, if you have a class 2 compound, exposure will go up. If you have a class 4, we have learned, and I have learned for sure, that it is good to be careful with the confidence that you communicate to the project teams, because you might find some surprises. There are some data that suggest that BCS4 drugs will show enhanced exposure and presence of food, but in the Novartis chemistry space, we have had several cases where exposure is actually reduced. And here we have such an example. So this compound, it has very low solubility, has a high fasted and fat ratio, and you can see here, if you see these data and you just look at fat over fasted, this is a high ratio. So you would assume that if the compound is not completely absorbed due to solubility limitations under the fasted state, that when you have a fat state condition, because of this very favorable solubility dissolution, that you would see an increase in exposure. And what we had done is we had actually used preclinical data and GastroPlus to predict the outcome. And our message to the team had been there will be no positive food effect. And here the clinical data, what we saw, we certainly did not see a positive food effect, but what we saw was a surprise negative food effect, reduced exposure and presence of food, which was not welcome news by the clinical teams. And we then went back and looked at the rat BDDCS, and which we didn't have at that time, and we confirmed that in the rat the compound behaves like a BDDCS4 drug. So the message here is that the fat and fasted solubility ratio for class 4 compound may or may not point you in the right direction, and in this case it was completely misleading. And just to show the more preclinical data, you can see here in the dog we had the high bioavailability in both states, but in the human, of course, we had the reduced exposure. So the dog for this BDDCS class 4 compound was not predictive, likely due in part to the paracellular absorption, and they have more leaky tight junction. So we were frustrated for a while to figure out why we couldn't predict this. And again, GastroPlus and other models, they're very good, but it depends on the input parameters. So one of the area which is important is the biorelevant generation of input parameters. That includes solubility and also permeability here. And to make a long story short, we used an assay modified from this paper, where we are using now FASIF and FESIF media to look at the apparent KECO2 permeability. And you can see for this compound, if you look at the FASIF versus the FESIF, the permeability is significantly reduced. We think this is because of micellular complexation in the presence of bile salts, given the high lipophilicity of the compound. But we didn't have this assay when we were doing our original GastroPlus prediction. Therefore, for this compound, when we had done the original prediction, here you can see is what we had predicted, and here is our outcome, as I already pointed out to. So we concluded that the KECO2 permeability that we had input into the model was not biorelevant, and therefore we did not anticipate this negative food effect. However, of course, if you input now this 8-fold, which was a ratio of 8-fold here, FASIF and FESIF, if you include this into the model, you can see that the model now can describe both the fasted and the fed. This is not drawn to scale here. These are different time axes, concentration axes. So you can see that the 8-fold lower permeability could very well describe the negative food effect that we have seen. So with this, I think we can stop here and open it up to a few questions, since it's about 12 o'clock. Thank you.
Well, thank you very much, Tycho and Wen, for the great presentation. At this point, we will open up for questions. We already saw quite a few questions came in. Here's one first question. Slide number 11. Human Tmax was underpredicted, possibly due to large, and parenthesis, unphysiologic percentage of water volume of 40% for small intestine.
So if we understand the question correctly, this is about our inputs that we are using in the colon for the water content in GastroPlus. Is that the question?
The question is from Steve Sutton.
Yes, that's what the question seems to be, whether the Tmax, which you were showing on slide 11, seems to be a little underpredicted, whether that could be due to the high volumes of water in the intestine, whether you explored that effect.
Yeah, actually, we tried the 10% of water content in small intestine, the 1% of water in colon, but somehow we failed to reproduce observed PK profile in the dogs, so that's why we still went back to use the default water content percentage provided by GastroPlus. And actually, this compound shows very variable PK profiles in the humans, so largely GastroPlus can still predict the Tmax within the observed Tmax range in humans.
Okay, thank you. On the next question, for the first compound's human PK profile prediction, the concentrations in the predicted versus observed plasma profiles were very low. What was the selected dose? Was the model also tested at higher doses?
Yes, actually, for the graph present here, the dose was very low, at about 1 milligram in human, and the model can successfully predict the observed PK profiles up to much higher dose, like 50 and 100 milligrams for this compound.
Okay, thank you.
I think it's safe to say that when we do the human PK prediction, we usually look at multiple doses. The reason is because when we do the initial assessment prior to first in human, it's often possible that during the SAD-MAD part that a wider dose range has to be covered. So we will usually communicate to the team a dose range and the performance over a dose range that should cover more the paradigm that's used in the SAD-MAD part.
Okay, thank you. The next question, why was drug U originally classified as BCS2 with moderate permeability? Was in vivo absorption good? That is larger than 90%?
Yes, actually, so for the drug U, the compound had a good bioavailability fraction absorbed for low doses. And so it didn't, the moderate permeability was not really a flag. So that's where, when we're looking at permeability, that's sometimes where we run into surprises, right? Because high permeability, at least according to BCS, is hard to get. So you would need to have a fraction absorbed, or a fraction absorbed above 85 or close to 90%, or you would have a Keiko2 permeability that's higher than propanol or metoprolol. But sometimes we have this moderate permeability compounds, and it's the moderate that can kill you. We're talking about can kill you in terms of this modified release assessment that Wenlin talked about, but apparently this moderate permeability can give you other issues. And for this compound, we did not anticipate the issue with this complexation because of this very high lipophilicity. So the reason we didn't know it was a more behaving like a BCS class 4 drug was because we thought moderate permeability is high enough, but also because we didn't have the metabolism information.
Okay, thank you, Tycho. The next question. Can you explain the biorelevant P effect again? I think the details are mixed here.
You're talking about the biosalt effect, right?
It's asking to explain the biorelevant P effect, the permeability.
Oh, okay.
The effective permeability.
Or the effective permeability, okay.
What do you mean by biorelevant P effect? P effective.
Yeah. Okay. So what we're talking about, when you use a model such as GastroPlus, you're inputting a permeability that you feel is relevant for the human situation. Now, because when you run traditional Keiko2 assays, they work quite well. You have pH 6574 or 7474, and you normally use some HBSS type of buffer. But those buffers, they may work for the majority of the compound. But for drug U, this type of buffer is not apparently biorelevant. So what happens here in the fed state here is that the compound, because it has bile acids and taurocholate, is complexed into these micelles. So the compound may be in solution, but it is not available to be absorbed to cross the intestinal membrane. And this is a little bit described in this paper here, and I did not put their examples here in this slide presentation, but this team found a similar trend that for compounds that are highly lipophilic, the permeability can change because in the fed state, the compound is completely emulsified and completely there is no driving force to cross the intestinal membrane because they're in these lipid or micelle particles.
Thank you. The next question, what is your evidence for using GastroPlus rather than absorption PBPK, rather than other software, for example, SimSip, for absorption PBPK modeling?
Yes, that's also a good question. At Novartis, we use GastroPlus. We have been using it for the past 10 years. And just in general usage, we know that DMPK is using it, and also our formulators is using it. Now, we are part of this European Orbital Consortium, and Simulations Plus is also part of this. And part of the Orbital Consortium, Orbital stands for Oral Biopharmaceutics Tools, is to look at the latest softwares and input parameters and figure out what models can be used under what condition and what are the best input parameters. At Novartis, we use the softwares both because they have different strengths. we use GastroPlus for all preclinical to clinical outcomes, and also for supporting tox studies, including even for PK/PD related studies. We do use SimCip in cases where there's already a DDI model available, and where we are interested in specific organ impairment populations or other specific populations. So to us, the strengths of the softwares are actually complementary.
Thank you. Have you done repeated dosing in any of the case studies?
Repeat doses, yes. When we look at our dose predictions for human, or any clinical predictions for that matter, we usually look very closely at all of our tox data. And tox data means day 1 versus, for example, day 25. If we see a species disconnect where, number one, after multiple dosing, in one species exposure goes up, in another species exposure goes down, we have a problem, and we tell the team that there are 2 scenarios that are possible. We then use this information to describe the pharmacokinetics in the rat or in the dog. If there's a mechanism such as induction, then that can be incorporated. But if we don't know the mechanism, we still use the information from both species and use it in the human PK prediction and give the team a range. Because to us, the human PK predictions are important to identify the risks, and majority doesn't rule. In other words, if you have mouse PK and rat PK which are in agreement, which do not agree with after multiple dosing with the dog, you cannot say I'm going to throw the dog out unless you have maybe hepatocyte or other data that says that you can do that. But yes, we always look at the multiple days, multiple dose data, and we also look at various doses, especially in the tox studies, because higher dose studies can sometimes tell us also more about the solubility of the compound.
Okay, thank you. The next question, could you comment on how the in vivo relevant deconvoluted solubility is obtained?
Yes. Let me go back to an earlier slide. In this paper, we discussed this concept a little bit. I think so. The way we will do this is we will first of all use animal data, which uses a dose that is in the human relevant range, because you know that in tox studies often you go much higher. So if we want to find out the in vivo solubility that is relevant for the human situation, we typically look at what is the what animal group or animal PK study used a dose that is relevant and translatable to human. And if you don't know the factors, they are in the FDA guidance on maximum recommended starting dose. But for example, for the rat, the dose is usually 6 times higher on a milligram per kilogram basis. So if in the human, if you have 1 milligram per kilogram, that's equivalent to 6 milligram per kilogram in the rat. Just talking about the maximum recommended starting dose. So we will figure out what the animal data are relevant to the human range we're trying to cover first. And then because we have here, on this diagram, this is drawn for human, but you can use the same diagram for animals, because we will have usually the animal IV profile. Because we have the animal IV profile and we have the animal oral profile, and we usually have more than one dose group, usually have 2 or 3, we can then nail down the intravenous parameters. We have multiple dose groups for the oral arm, and then we can use the GastroPlus optimization module to tell us what is the most likely solubility that could fit the data. The method is not perfect, but it seems to work overall. So the key here is you have to look at multiple dose data, and if in doubt, you have to use the dose group, the dose strength that is most relevant for the human situation to give you an in vivo deconvoluted solubility data.
Okay, thank you. The next one. You showed the applying biorelevant media corrected the permeability improves prediction for food effect. Have you applied this to other compounds? Is this compound specific, or more universal?
We can say that we have used this assay now for about 10 compounds, and we have used this assay to include compounds where we know that we do not see a negative food effect. Because we don't want to use an assay that says you have a negative food effect for all compounds, right? That would not be discriminating. It is safe to say that when you have compounds where you see a positive food effect, or compounds where you see no food effect, this assay will still work. It will for compounds where you see a negative food effect. This reduction of permeability in the presence of fat state will be very pronounced, at least 3 to 4-fold.
Okay, thank you. The next, actually, there's two questions together. For the 18 compounds which Wenlin mentioned in her talk, are those compounds highly metabolized? The next question is, when you did the modeling, did you also try PBPK option instead of compartmental model?
Okay, so the first question, among these 18 Novartis compounds, it has BCS 1, 2, 3, and 4 compounds. So not all these compounds are highly metabolized. And for the second question, did I use the PBPK model to predict this, actually basically the distribution of the compound in human using the PBPK model? Actually, for all these compounds we use, majority we use a compartment model.
Okay, thank you, Wen. The next question. Did you have any experience of species differences in terms of food effects prediction that could be explained by solubility differences due to the different biocomposition observed between animal species and human?
Yes. For species differences in terms of the food effects, for compound U, we didn't show the data for the rat. In the dog, I think we can go back to that slide. We had no food effects, but in the rat, we had a positive food effect. Let me maybe see if I can go back at least to the dog data. Again, we are not showing the rat data here, but I can tell you in the rat, the absorption in the fasted state was not complete, and therefore we saw a positive food effect in the rat, but we did not see a food effect in the dog. So we have seen this type of disconnect, but we don't see that disconnect a lot. One other important thing here is, if we want to predict the food effect most reliably, there is an obvious circumstance that you need to take into consideration. The formulation that you're giving to the dog, we like to use dog for that, has to be closely resembling what you're giving to human. And this may be an obvious point, but sometimes it gets forgotten, where we may be evaluating a drug's formulation in a food effect early on, but then when you go in the clinic, the formulation has changed. And we have seen on several occasions that if you use a non-optimized formulation and test it in the dog, you can see a positive food effect, but then when you have an optimized formulation, you see no food effect. So it is important that for some drugs where the food effect is formulation dependent, if you want to predict the outcome in human, that you use a relevant formulation and a relevant dose in the dog to translate to human.
Thank you. This next question is about animal welfare. I guess they care about the human safety also. Eye pill contains metal component. Can dog or human remove it from the body after dosing?
The eye pill that we tested, when we dose them to dogs, they are readily recovered. And what you can do is, because there is a temperature sensor in the eye pill, you can also find out on the computer when it was eliminated, because the temperature will no longer be 37 degrees. So all of the capsules are recoverable in our studies. And there was a human study that was published. We should mention here, about 3 or 4 weeks ago, the Mediometrics team also published on human gastrointestinal pH values, and that if anybody is interested in human modeling and they find out in GastroPlus that they cannot describe data, you might want to look at that paper. We were not part of this paper, but I think human relevant pH data is important and it's hard to get. So again, the Mediometrics folks, they have measured it in human, and these capsules are recoverable. We find no toxicity.
We still have 7 or 8 questions in the queue. We need to speed up a little bit, or we'll have to extend our time limit. The next question is, does the model take into account or predict the nonlinear PK processes?
Non-linearity is very difficult to readily include. We do include non-linearity more easily when it's relevant to solubility dissolution. But there are other cases when you see dose over proportional PK, and that's where for example, if you find dose over proportional PK in the rat, how do you now translate this to human? This translation is notoriously difficult. For example, in this publication that Wenlin talked about, there's one compound we didn't predict very well at one dose strength. I don't remember now which one it is, but it's one that wasn't predicted very well at the 100 milligram dose. But at 10 milligram, the prediction would have been very well within 1.5-fold. And so the reason we predicted quite well for the lower dose was because the rat parameters were relevant, but there were nonlinearities in the rat, and we couldn't scale this properly to the dose that we list in the paper, and we were significantly off. So when you have the nonlinearity, all you can do is look at all your tox data and see if you understand the mechanism of the nonlinearity and include it in your model. And you can use GastroPlus to do this, but the magnitude is still, in my mind, fraught with some uncertainty.
If I could get just a very quick note to follow up on that, as Tycho just mentioned at the end, you can account for it within GastroPlus. You have the capability, but the major issue is with the interspecies differences. So prediction from animals to human, and the problem is mainly because unknown differences in the enzymes or transport expression levels and so on. The capability is there, but some of the physiological information is still missing, so that limits the predictability.
Thank you, Vera. The next question, what was the log D, log P of the drug U? what could be a flag for biosalt micelle capture?
The log D was very high. We say, I think we say here it's larger than 4, but it was very high and very lipophilic compound. The other thing we can tell you that it might be bringing up a flag to anybody who is working in this area is if you run KICO2 assays and all of a sudden you have very low recovery, or you have adsorption, meaning you have a stickiness of the compound. In my mind, these things could be bringing you in a situation where in the fed state, because you have these bile acids, all of a sudden they're just trapped. So very high lipophilicity might be a trigger here.
Okay, and he asked the next question, the same individual. For drug U, did you evaluate the effect of selecting, deselecting the adjusted diffusion coefficient for biosalt effect option?
We did not do that part because we didn't have any data to put into the model. You see, this is the beauty of tools like GastroPlus is you can do parameter sensitivity analysis, and I'm sure you can mathematically do this. Again, we haven't done it, but I'm sure it could be done. But then how the challenge is you have to have some in vitro data from your formulation lab or other data to justify why you're changing the diffusion coefficient. But the diffusion coefficient certainly could be used as a surrogate instead of KICO2 permeability to probably give you the same type of mathematical effect.
Thank you. The next one, can alcohol induce the dose dumping be predicted using GastroPlus? If yes, does it give any more information than the typical in vitro dissolutions in presence of, say, for example, 40% alcohol in 0.1 normal HCl?
The dose dumping is a topic which is relevant for, you know, when you're concerned about the toxicity of the compound and other things. We usually do not get a lot of questions about what happens with dose dumping because we only have a few cases where we're really heavily working on the modified release formulations. So maybe, I don't know, Vera, if you have some ideas for this dose dumping question when you have this issue that comes up.
Yeah, in theory it could be done, as you mentioned, you know, doing the sensitivity analysis and things like that, but we have not done extensive studies as comparing whether the in vitro dissolutions in the presence of alcohol would give you a relevant answer or not. I guess my answer right now is that in theory you could do it, and possibly the sensitivity analysis would be one of the good tools to use to evaluate what kind of issues you might be dealing with, but we have not had enough cases to actually comment on how the in vitro dissolutions would be predictive.
Thank you. We have 2 more questions to go. The next one, when you use allometric scaling, and that is the Wajima method, did you predict not just for drugs with one compartmental profile, but also 2 compartments? Is there a situation where one animal species shows 1 compartment PK, but the other species showed 2 compartments? If there is, how to deal with that?
Okay, this is a very good question. Actually, we often encounter this species disconnection, that one animal species shows 1 compartment, another species shows 2 compartments. Under this situation, we need to use our experience and knowledge to select the appropriate compartment for human. For example, so if the dog or the monkey is pharmacology species, so for sure we would prefer the dog pattern to predict the humans and other animal species. And another way is usually we we conduct the population PK analysis for even for the preclinical study, because sometimes the conventional PK analysis cannot quite well determine which compartment model can best describe the PK behavior of the compound in the preclinical animal species due to very limited number of the animal tested in the IV study. So that's why we can use population PK approach to facilitate the selection of the compartment model for the human.
And maybe also adding to this, if you on this is the Vajima plot, right? If you have a species disconnect, for example, that your intravenous clearance profile is very different between dog and rat, and if the species disconnect is related to disposition parameters, then in this plot you will see the curves will not superimpose on each other. So you can use this Vajima analysis to identify a species disconnect quite well. And then unless you have IVIVC as in IVIVE, meaning hepatocyte data, then you will identify there's a risk here. In some cases you may be able to correct for that and say, well, maybe the dog is more relevant or the rat is more relevant to human, but at least in the Vajima plot you will be able to see if there's a species disconnect. So that's quite useful for that.
Okay, thank you. 2 more questions, and hopefully the answer are the brief. You mentioned briefly about using rat metabolism. Could you give more details on using rat metabolism data to predict the humans?
Yes. So the rat is a very good model for absorption. I think most of us we will agree on this. When you look at this paradigm here, which is this rat BDDCS, the metabolism cutoffs that we put here, they're a little bit different than the human one. And the human one that we refer to here is from Leslie Bennett. Leslie Bennett had originally, I think, maybe these numbers, or might have been slightly different, might have been more than 50%. So what we do is when we do the very early human PK assessment, we look at first with the hepatocytes, rat and human hepatocytes. We look, is there any very significant difference in the metabolism pattern? But for a lot of the compounds, we just look at the totality, and we just look at how much drug is found intact in the excreta. And we use this information also for our DDI assessments, because in DDI assessments you need to know how much of the drug is metabolized versus how much is left intact. Now, do we know that in 100% of the cases that the fraction metabolized in the rat is identical to that in the human? No, maybe not for BCS class 4 drugs, but for if a compound is highly metabolized in the rat, typically it's also metabolized in human. There may be different metabolites, but the fraction of drug left intact is often similar, at least based on our analysis.
Okay, thank you. Last quick question. Can the effect of different types of food be predicted using GastroPlus? That is typical foods from different global geographies.
So I'm going back to Dr. Wendling's slide. Okay? So you can see here. So the quick answer to the question is, it's not all that easy, but our attempt is looking at the food effect, and as you know, there's a high fat and then there's a low fat meal that sometimes are used for the assessment. For example, in oncology, you may not always give with high fat, but also with called low fat. And low fat, by the way, is not defined yet in the food effect guidance, but it will be defined in the new food effect guidance that will come out. Now, when you look at the low fat versus high fat, maybe one way to deal with that is to look at these biosalt concentrations that is released with a high fat versus a low fat meal. Now, Dr. Wendling and I recently had a discussion because our thinking was, well, if we know the biosalt concentration with a high fat meal, and then we go to a low fat meal, which is typically defined, at least at Novartis, as about half of the calories in terms of fat, then could you halve the numbers? So that is not completely validated and verified. And by the way, we also don't see usually half the food effect for a low-fat meal compared to a high-fat meal. So there's definitely some non-linearity there. So even though you give only half the fat calories, the food effect is not half with the low-fat meal for some of our compounds. But if we had to guesstimate, what we would do is we would change this biosalt concentration here in GastroPlus to have different concentrations. This is fasted, you can see here. The fat one, I think, has a 13.8 in this number here. So if you're going to low fat, you may be able to play with these numbers, but maybe Vera has some other ideas.
No, that would be actually, sorry, John, that would be actually my recommendation as well. The only the one way to account for it right now would be maybe changing the biosalt concentrations, and the other aspect may be also the stomach transit time, right? Because the gastric emptying is going to be in the fat state, is driven by the calories. So the stomach is gradually releasing certain number of calories per hour or per time. So with the differences in calories, the stomach transit time may be affected as well. But you know, as for the biosalts, how much that would be affected by the high-fat or low-fat meal, that is still a bit questionable. One of the questions is how much would the fat coming from the meal be affecting the solubility or dissolution of your compound, versus how much is the contribution of the physiological bile salts that will be in the intestine anyway. So.
All right, we covered all the questions we've received. We are a little bit beyond our time for this webinar. Again, I would like to thank Tycho, Wen, and Vera for answering these questions. For the audience, thank you all for your participation. You may now disconnect from the webinar. Thank you, everyone.
Thank you. Thank you all.